To finish the chapter on the immune system, we're going to finish thinking about adaptive immunity by concentrating on what antibodies are. The section on lymphocytes ended by looking at how B sites are activated. When a B cell receptor, which is a type of antibody, binds to its particular antigen, that antigen is internalized and presented on the surface of the B cell with MHC2 so that a CD4 positive helper T cell will recognize it and activate the B cell. Let's look a little bit more closely at what antibodies are, and also we'll look at a related molecule called the T cell receptor, which came up in the lymphocyte section, but we're going to see how it fits into the story a little bit better here. Antibodies are composed of 4 protein subunits. 2 of them are called heavy chains and 2 are called light chains. Heavy and light refers to basically the length of the protein subunit. Heavy or longer proteins and light are shorter protein. The proteins are separated out into loops, which are represented in the schematic picture by the segments. And in the ribbon model that we can see here, you can see the different loops kind of clustered together. The first loop out at the end of the Y shape of an antibody is the variable region in either the light or the heavy chain. And then the other 3 loops in the heavy chain and the 1 other loop in the light chain are the constant regions. The variable region is representing a part of the gene that gets transcribed and translated into protein from a randomly sorted region in the DNA. When each B lymphocyte is originally generated, that part of the gene randomly sorts itself to make a unique version of the antibody genes. And then those are used to make the light and heavy chains that become the antibody. This way, each individual lymphocyte only makes antibodies against a particular antigen, and there's a unique antigen binding site at that region on the antibody. Where the light chains end, and we just have the constant parts of the heavy chain, what's labeled here is the FC region. There's a hinge region, a very loose connection between the 2 parts so that the arms, if you will, of the antibodies can kind of go any direction they need to. The book begins by introducing the T cell receptor, which we see here. It's very similar to an antibody in structure, except that it only has 1 pair of subunits called the alpha chain and the beta chain. They have a variable region also, just like the antibodies, but resulting in only 1 antigen binding site. And then there's a constant region and the transmembrane region, which would effectively be like the constant regions of the antibody. These are just found on T cells as opposed to related to B cells, but they still interact with specific antigens the way that antibodies do. This is another representation of what an antibody looks like. This was a figure that I used in the presentation about the blood, talking about the plasma proteins, which is one place where we find immunoglobulins or antibodies. This figure from the book is showing how T cell receptors, and antibodies for that matter, interact with antigens. The book starts by introducing T cell receptors before antibodies, and so as they're describing this interaction, they're using T cell receptors. But this is probably not a very realistic representation of how this would interact with T cells. So imagine these are antibodies. So the antibodies have the antigen binding sites, which bind to specific antigenic determinant spots, specific shapes on the protein or whatever the antigen structure is. So here we see 3 different things binding to it, represented by 3 different colors, because they all have unique antigen binding sites binding to unique antigenic determinant parts of the protein that's the antigen. When an antibody and an antigen interact, when they form a complex, here's a list of the different things that can happen. Now, Plano is a way to remember this, a mnemonic to keep these different things straight. P stands for precipitation, which is what happens when small antigens, meaning proteins, are all linked together by antibodies. They won't dissolve in the bodily fluid where they're found anymore, and they'll precipitate out of solution. In that situation, then, they would be able to be cleared away by phagocytes. Lysis is referring to complement, and so the classical complement pathway that depends on antibodies interacting with complement protein 1, leading to complement proteins 2 and 4, forming the C3 convertase, that's lysis in this case. Agglutination is when large antigens, meaning cells, are linked together by antibodies. antibodies. And it's very like precipitation, just with a different size antigen. And we've talked about agglutination before when we talked about clumping of cells when we were testing for blood typing. That's essentially what that refers to. Neutralization is when the antibodies are covering the antigen so that it's no longer effective. We usually associate this with virus particles. When they get covered in antibodies, they can't interact with the host cells and cause an infection, so they're inactivated. There are other things that could be neutralized too, but virus particles are the best example of it. And then finally, O stands for opsonization, which is any increase in the phagocytic activity to clear away the antigens. So with complement, C3b actually increases this itself. It's just another example of opsonization. But antibodies themselves, whether they're involved in the classical complement pathway or not, can opsonize antigens and help clear them away, help the phagocytes find them and destroy them. Now really, that's the end point of all of these things. Precipitation, lysis, agglutination, neutralization needs to lead to all of those antigens being removed by phagocytosis just the same. But opsonization is when it's the only thing that happens. There are 5 types of antibodies. Now, any given B lymphocyte is going to make antibodies of a single sort in that they only recognize one antigen. But there's 5 different variations in what they look like and how they behave. So this table sort of goes through those. To begin with, the most common one is the IgG. Ig stands for immunoglobulin, another word for antibody, and then this is class G, which stands for gamma. It's the most common. About 80% of the antibodies in your system are that type. One very important thing about what IgGs do is they cross the placental barrier, which means that a mother will share her immunity with the embryo in utero, so that the baby has some adaptive immunity, though passive, for when it's newborn. The next most common type is the IgA at about 13%. It's often found in a dimer form, and it's secreted in most bodily fluids. So in the mucus and tears and saliva associated with the barriers of the innate system. Colostrum is referring to early breast milk, and this is another way that a mother shares her immunity with a newborn. Next most common is the IgM, which is best known for configuring itself into pentamers, so 5 separate antibodies all linked together, giving this 10 different antigen binding sites. And it can cross-link a lot of different things together. It's very good for precipitation and agglutination in that regard. Next most common is the IgD, which is a monomer that's often associated with the B cell receptors, such as on the naive B cells. And then IgE is a very rare type, just a fraction of a percent, but it's associated with mast cells and basophils. Mast cells, when they're activated by an IgE that's acting as a receptor on the surface of the mast cell, will cause an inflammatory response, usually in terms of an allergy, not an actual immune response. Basophils are very rare types of white blood cells, but they're best known for being important for fighting off parasites, specifically parasitic worms. There is an interesting hypothesis that ties parasitic worms and allergies together, which is the focus of the podcast that I referred to back in the pathogens section of this presentation. In this figure, we see activation of B cells, but also the idea of primary and secondary responses and the effect of immunological memory. So we see 3 naive B cells here, each with their own B cell receptors, depicted in different colors, blue, red, and purple. Only the red B cell receptor, or antibody, recognizes this particular triangular-shaped antigen. When it gets activated by that, when it recognizes that antigen, it becomes activated, which does involve the helper T cell, which is not in this picture. The cell then divides, differentiates, makes memory B cells and plasma cells. Plasma cells look a little bit different than the B cells here because they have rough ER and Golgi apparatus to make a bunch of secreted proteins, which are going to be secreted into the plasma. The memory B cells are available so that a secondary response can happen very quickly and very strongly, so that there's no need to wait for activation. This graph suggests the primary and secondary immune responses laid out as curves representing the concentration of antibody. After the initial exposure to an antigen or pathogen, the primary immune response will build up antibodies to fight off that infection. The level won't quite go all the way back down to its initial level, but then with a secondary exposure, there'll be a very rapid increase for the secondary immune response to fight off the infection even quicker and probably keep symptoms from setting in. I wanted to add showing you this graph because it makes an important point that the previous graph actually sort of left out. First off, this is the classical representation of that primary-secondary immune response graph. It compares the non-specific release of IgMs from B cells, which is constant every time you get an infection of some sort, versus the very specific release of IgGs between the first time that the lymphocytes are activated and then every subsequent exposure which the memory B cells are available for. But what I wanted to point out here is on the y-axis, the concentration of antibody, the antibody titer, is actually in a logarithmic scale, which is to say the primary versus secondary response are orders of magnitude different. So do understand that with the secondary immune response, we don't see symptoms because there's such a strong reaction thanks to the memory cells that are there. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. Opsonization is the term for when phagocytic activity is increased. In terms of antibodies, when antibodies are attached to the antigen, it makes it easier for the phagocytes to find what needs to be ingested. Other substances can do that also. We've also seen in complement, the C3b fragment can opsonize as well.